Keep the check. So I'm not going to wear, look, I'm very exothermic, and so I kind of give off heat but I was told I had to come in looking smart, at least. So there we go. Hi, guys. I'm here to give the Haldane lecture. So this is an interesting chap. I'm sorry, I'm going to kind of wander free range out here for a bit, if that's okay. John Burden Sanderson Haldane, FRS. So, and he was a British and then latterly Indian geneticist. Okay, and actually he started life as a mathematician. I might be getting some of these things wrong, so hopefully not for that. And amongst the many things which he actually has done was he did, for example, I just picked some of these, Wikipedia, is that he actually demonstrated the first time genetic linkage in mammals. Okay, he came up with the sickle cell malaria hypothesis. So this is when he actually observed that, hang on a second, how come, people with sickle cell disease, are actually found more commonly in areas where there's malaria, and as it turns out now, if you're a heterozygous carrier for sickle cell, it's actually protective against malaria. Someone else actually demonstrated it, but he came up with that hypothesis, and okay, an inordinate fondness of beetles. He doesn't particularly have an inordinate fondness of beetles, and this might be an entirely apocryphal story, to be fair. But apparently, and when being questioned by the clergy or something about what he thought about the divine thoughts, what the creator might have been thinking, and he said he had an inordinate fondness of beetles, because actually the vast, a huge proportion of life on Earth are actually beetles. I think there are like 400,000 different types of beetles on Earth, so there we go for that. But finally, he was also a huge and passionate, not only supporter and advocate for, but also a huge and passionate science communicator. And so I am deeply honored to the Genetic Society, for giving me this opportunity to give you the lecture, the 2019 Haldane lecture to all of you. And thank you, all of you, who I think you need to be questioning your life decisions while you're here on a Friday night. But that's okay. We'll deal with that. Maybe you'll be doing it less. But I am deeply honored. And I'm here to actually speak about obesity. And I'm posing the question, okay, which I do think it's relevant because of where we are today, but hopefully it's a provocative question. Is obesity a choice? In fact, I'm going to ask the question. There'll be some audience participation tonight, so I'm going to warn people. How many people here think that obesity is a choice? Okay, this is good. Although it does look like a largely friendly audience. Why? But okay. So let me. I don't think it is. But let me give you. It's a caveated. I don't think it is. And hopefully, at the end, I either have convinced you or I have not. I'm based at the University of Cambridge, and I work at the MRC Metabolic Diseases Unit. Now, I'm a geneticist by trade, by its very definition. Otherwise, I wouldn't be here with the Genetic Society blessing. And I think that's a perfectly upstanding thing to do. My mother-in-law still speaks to me. So this is all very, very, very good. But when people ask what disease I study, because people often use genetics in order to understand disease, because how best to understand how something works than when something doesn't work. And I say obesity, actually I say body weight, obesity being one end of the spectrum, immediately I become the bad person. And I become the bad person because I'm perceived as giving fat people, overweight people, people with obesity, none of the terms which I use in any pejorative sense whatsoever, an excuse, which philosophically to me has always been an interesting take, I think, on and now. Because if I was studying the genetics of dementia, the genetics of rheumatoid arthritis, the genetics of cancer, would I suddenly be giving those people suffering from those particular diseases an excuse? I'd like to think not. I'd be trying to understand a pathway, I'd be trying to understand a mechanism, I'd be trying to fix a problem. But yet, you speak about body weight, and suddenly you're giving people an excuse. And I think a large part of it is down to this. Everyone has seen versions of this everywhere. This is the scales of justice. This is otherwise known, for those of you who've done GCSE physics, the first law of thermodynamics. Okay, so you can't magic calories from anywhere, and you can't magic the calories away. So you got to eat more than you burn in order to gain weight, and ergo the only way to lose weight is to burn more than you eat. Okay, so there we go. You guys are thinking, did he just say eat less and move more? Did I just pay money to hear that? Yes, you did, but it's true, and this is the thing. There is a huge but at the end, but it is true because it's physics, and we're not going to magically get past physics because it is physics. The question, however, is not how we get to the body weight we are, which is going to be down to physics because that's the only way we're going to be able to get the calories in and get the calories out. But it's why. Why do people behave differently around food? So let me give you a scenario. This is the boardroom scenario. Okay, so you're in a meeting on a Monday. It's 8 freaking a.m. in the morning. Okay, and so you're sat there, and suddenly someone slides a plate of cookies into the middle of the table. Okay, now I'm going to argue there are 4 different behaviors to this sliding plate of cookies. There are going to be some people before the plate even stopped sliding. Okay, would have already reached for the cookie and had one. Okay, you know who you are. Okay, now. There's a second group of person, okay, who longs for the cookie, dreams of the cookie, but for whatever reason does not take the cookie. But the end point of this, dreaming and longing of the cookie, is they don't listen to the meeting, right? So the entire meeting is gone, and they've been thinking about the cookie the entire time. You know who you are, okay? So now then there's a third type of person, the most annoying. I want to argue, are the people who don't even know the cookie has arrived, okay? This guy comes in, they're going, what cookie? I didn't know there was a cookie. And then there's the fourth type of person in whom they eat the cookie without even knowing the cookie is there. John, did you like the cookie? What cookie, right? And I think you know who you are too. Now those 4 different behaviors, are they willpower? Is it down to moral fiber? Is it? No, because a large part of it, yes, okay, there's going to be social, there's going to be cultural norms, but this is a situation in which you're in the same office, you're sat around the same table, you're broadly of the same socioeconomic class, broadly, right? You're office workers, whatever, and you're actually doing what you're doing, and yet you behave so differently around food. And a large part of that is going to have a biological basis. And when you read biology, you're going to have to say genetics. And a large part of it is actually going to be some genetic basis. So the question is not how we get to how we are, which is going to be down to physics, yes? Okay, and let's all accept that. But why do people eat more? What drives someone to eat more and therefore gain weight? Or what drives someone to not eat more and therefore be either average weight or skinny or what have you? And there we have my particular interest in in my field of study. Why are people different sizes in exactly the same food environment that we're actually currently living in at the moment? Now, we know that there's a huge problem with obesity at the moment, okay? I'm not going to harp on about this particular figure because I think it's well rehearsed, you can barely turn on a television, to have a documentary made about it. I'm responsible for some of them, so I understand that. But only to say that while the fact that in Western Europe, in North America, what have you, that there is a serious problem with obesity, type 2 diabetes, and all of the other comorbidities, yes, very well rehearsed, we're actually nowhere near the maximum of the problem. Because of this tiny corner of the world over here, okay, the Asia, okay, so the Indian subcontinent and China, okay, where, fine, there's probably going to be, roughly speaking, 300 million mainland Chinese people with enough expendable income to go on holidays where you guys go on holidays, okay? And same number probably of South Asian Indians, okay? But you have to remember, there are still 1 billion people, okay, stuck out in the sticks, both in India and in China, in where, if you're actually poor, okay, there's not enough food and you're skinny, okay? It's a bad thing. It's a bad thing. Unlike clearly in the West, where lower socioeconomic class is actually linked to a greater risk of obesity. So while their governments are trying to drag their populations, out of poverty or what word you want to use, suddenly they're going to be exposed to the same environment that we're being exposed to. When an additional 2 billion people in the world are suddenly exposed to this, we will begin to reach the maximum of the problem. And I'd like to think we could try and fix this before we actually get there. Now, the one thing about talking about genetics and about obesity is that people say, well, our genes haven't changed. What are you talking about? How can that be the case? And it's true, okay? So this is data from the US Center for Disease Control, but I stole the GIF from the Washington Post, okay? But here, and if you can Google this and you can actually get the GIF as well. So this, I love this. This is like a nerd's, dream. It's like a 6D, graph. It's got time, it's got, BMI, it's got sex, it's got frequency, and it's got a histogram. We love it, okay? But what you see, so now BMI is imperfect. It is imperfect. But let's go with the numbers we know, okay, where between 20 and 25, you're normal body weight, right? And then between 25 and 30, you're overweight, and above 30, you're obese. Once again, imperfect. We can discuss this later, but let's go with that. Then you can see, let's go back to 1984. We can see that the average body weight of a male, okay, in 1984, or BMI, pardon me, was 22 to 23, so normal, okay, in 1984. Whereas in 2014, you can see that that has risen to the overweight category of BMI of 27. Now, between 84 and 2014, now I certainly, and I look in the audience, I think, sadly, the vast majority of you were alive throughout the entire process, like me, okay? And we're here, and our genes haven't changed. We haven't suddenly evolved a second head, okay? You know, when we're there. So it's correct that the reason why we're different sizes now is because of the change in environment, because our genes haven't changed. Okay, so that's true. But, and here's the but, if it was all a response to just the environment with no biological input, then the histogram from 1984 should, in theory, shift in its entirety upwards so that you actually get a shape that kind of looks like the Golden Gate Bridge, okay? But that's not what you see. You don't see a shift in the entire curve, you see a change in the shape of the curve. Now, assuming that everyone in 1984, okay, well, is still alive now, okay, right? Then the only way there's a change in shape is if every data point in there, every human being in there, behaved differently when the environment changed from 1984 to now. So sure, why have we as a population, as a species, become heavier? Because of the change in environment, our life, our exercise, our jobs, the kind of food we're exposed to. Okay, the entire environment. But how obese we become, do we become overweight, do we gain any weight at all? That is powerfully biologically influenced and actually has a huge genetic role. And we understand this because in part, these are the Ventalican twins, for those of you who know them, in part from twin studies. Okay, so why study twins, you guys might actually ask. Well, as we all know, they are identical twins, and they share 100% of DNA material, so all the genes they share, right? And then you have fraternal twins, non-identical twins, and they would share as much genetic material as you would with your own siblings or, for that matter, your parents. 50%. So you could take any given human trait and ask the question, well, what happens when I share all my genes versus with a trait versus when you share 50%, and work out what we call heritability. Now, heritability is the percentage of a variation of a trait, which is down to your genes versus the environment. Okay, let me give you some examples. If I had hair, my hair would be black. And I want to argue that hair coloration is very powerfully genetically influenced with very little environmental impact. Bleach does not count, ladies, okay? Now let's look at another trait, freckles. Now freckles are clearly also going to have a genetic influence, clearly, but whether or not you get freckles, how many freckles you get at all, is going to depend on whether or not you wear t-shirts. Do I like to stand in the sun? So then we have a genetically influenced trait with a powerful environmental impact. As it turns out, every single human behavior and human trait will have a genetic influence, every single one, even car accidents, okay? But the trick and the trickiness is what role does the environment play? Okay, so it's always the two together. There's always a genetic influence. And if you do that math, then what actually happens is you actually see that there is a 70% heritability of body weight. So it's not zero, by any means. And it's not 100% either. Just to give you some perspective, the heritability of height is 85%. Okay, so it's not as high as height, but none of us here would debate that height has a genetic influence, okay, but it is certainly approaching that of height. And we now know, okay, of several hundred genes, and we'll come and actually talk about a few of them, which actually influences our behavior around food, and even whether or not we like certain foods or not, therefore influencing whether or not we might actually choose something. Genetics and feeding behavior, really? Okay, so more audience participation. Okay, so Peter is going to give me, where's Peter? There we go, is going to give me a hand here. I need 6 volunteers. It's terrible. I know it's a terrible thing. Who wants to You've just been volunteered. So who else wants to? I need 6 volunteers, ladies and gentlemen. Come on, guys, don't be shy. So that's 1, that's 2 over here, that's 2, 3, the lady, the 4, that's 4 ladies. I need 2, are we doing, you're down out already. Would you want to participate as well? Okay, 5. I need a gentleman. Okay, you. There. Sorry. Sorry. Right. Okay, so stand in a row. So bear with me here, ladies and gentlemen. Okay, so stand in a row over here. Now, I promise I am not going to poison anyone, and just to show people that I'm not going to poison anyone, I'm also going to partake in this particular experiment, okay? So now, everyone listen. You're going to get a piece of paper. Don't do anything till I tell you, please. Okay, so what happens is when I tell you to, I want you to put this piece of paper on your tongue and tell me what you taste. This is designed for children. So hopefully this will work. Okay, everyone, 1, 2, 3, put it on your tongue. 1, 2, 3. Do you taste anything? Good, there was nothing on there. Okay, so this is otherwise known, scientists out there, as the control experiment, okay? So now we know what a piece of paper that doesn't have anything tastes like, okay? Now this is the experiment. Please don't put anything on your mouth. Don't mess up the two, because otherwise. So on this next piece of paper, I have randomly put some chemical flavoring. It's not poisonous, okay? And I want to, in exactly the same way, now exactly the same thing, there'll be something to taste now. I want you to put it on the tongue once I do the same thing, and then I want you to tell me, raise your hand if you taste anything, okay? What? Okay. Okay, 1, 2, 3. Anyone taste anything? Oh, yeah. Okay, all right, good. Once again, I also lied. This is terrible. This is a terrible thing. So there are actually, there is, where's my pointer thing over here? Oh, there we go. So every one of you had the piece of chemical on there, and the chemical which I put on there is called phenylthiocarbamide. Okay, and phenylthiocarbamide is the bitterness that tends to be found in brassica. So, for example, in Brussels sprouts, in broccoli, and whether or not you can taste it, okay, is down to this gene called the TAS2R38 gene. And there's one single base pair change in there, okay, which allows I tasted it, but just FYI, but I've done this so many times I now can expect the flavor, okay, where that makes some of you taste the bitterness and some of you not taste the bitterness. So the ones of you who can taste the bitterness are called super tasters, okay? Now, you are therefore more likely, but not deterministic, more likely to dislike Brussels sprouts. So you're going to tell me, I love my Brussels sprouts, okay? But what I'm saying is that on a population, you can see that half of you here, roughly speaking, could taste it, half of you could not, okay? Everyone, I'm not white Caucasian, but you guys are all white Caucasian, and we're sat here, and so therein lies a situation with just one simple thing, whether or not the likelihood of you liking Brussels sprouts is actually down to your genes. Thank you very much, guys. Thanks so much. All right, so thank you. That worked. Yes, every time you do this, they're gonna come out and go, I hope they actually taste something. The kids, because I did the show recently in Hong Kong of all places. That's the kind of guy. That's how I roll. Okay, and the kids, because they're kids. Okay, like a 6-year-old kid. He put it in his mouth. You know. Adults, I thought you were remarkably well behaved. Anyway, so that is just one example. I just wanted to show you that it is, people think, oh, there can't be genes involved. Look, I'm just talking about whether or not you like Brussels sprouts, okay? And there is a plausible genetic information from there. But how about feeding? How about hunger? How about feeding behavior? Now, we now understand that there are pathways that actually control, or at least actually influence food intake and influence body weight. And the first real molecular handle that we ended up getting was by this molecule, which I'm going to spend a little bit of time on, called leptin, okay? And this mouse over here actually has a mutation in the leptin gene, and so therefore you can see it's more than twice the weight of its two littermates, its two siblings that is next door. So leptin, as it turns out, is made from fat, and it signals to the brain, okay? And so therefore it lets your brain know how much actually fat you have. So this is the way. So from there, and I'm going to be talking about the system in a second, from there we now have a general schema of how our brain controls food intake, okay? That they're going to be circulating hormonal and nutritional cues that circulate and then actually go to the brain, and your brain then translates and responds to these, and they're going to be genetic modifiers that actually run throughout the entire process. So some of these signals, so your brain needs to know two pieces of information in order to be able to modify food intake, or to influence food intake. It needs to know what you have just eaten and what you're currently eating, because obviously these are your short-term signals. And this involves this other thing here. Now, I don't know if any of you, some of you here would have ever seen a life-size knitted human gut. Well, it's your lucky day. Okay, I have the pattern for those of you knitting. So now plus or minus a couple of feet. So this is in here. It's the latest thing. So for this is otherwise known as the food to poop tube. Okay, and what happens is this is plus or minus a couple of feet, depending on whether or not you're 6'7" or 5'2", is how long your gut actually is. So this is the food side. Okay, now I'm supposed to kind of, okay, and then it's origami. So this bunch of pillows here are your internal organs, okay? And then you can see that this pink bit is actually your small intestine. And you can notice that the small intestine is not that small, and the big intestine is actually quite small. So the small intestine, you can see, and I won't stretch it out because it'll go past the cameras, and this is the poop side of things, and you can see. Now, and it's long. It's probably 8 meters, 21, 22 feet long, and it's all origamied into us. But what happens is as you're eating and the food goes down the esophagus and then into the stomach and then into the small intestine as it goes down, your brain is then informed of what happens, right? Not only in terms of how much food you're eating, not just bulk, but also the number of calories and the macronutrient content as it actually goes down. So how much protein, how much carbs, and it does this because as it goes down, it secretes hormones. The hormones go to the brain and says, you've had 200 calories, half of it is carbs, etc., etc., etc. So that's one piece of information, okay, that it has. But in addition to the short-term pieces of information, your brain also needs to know the long-term stores because it's unlike governments today, okay, you can't make decisions based on short-termism. You can, okay, but you also need to know what happens in the long term. And your long-term energy stores are going to come from your fat, okay? And this is what I want to spend the rest of the time to talking to you guys about. But that was almost like the what you ate sensing pathway. I want to talk to you about your fat sensing pathway that's actually here, which is actually part of in which leptin plays a key role because leptin is a fat hormone. And we now know, and this is the pathway I'll spend the rest of the time talking about, that the leptin-melanocortin pathway, leptin, I told you about that mouse that doesn't have leptin, comes from fat and signals to the brain. It signals to a class of neurons called the POMC neurons, and then it signals to the MC4 receptor, you heard of it in the introduction, influencing your food intake. And why we know that this is a critical part of your food intake control is because disruption at every single stage of this, not only in humans, not only in mice, results in severe feeding abnormalities and severe obesity. And I'll give you some examples to show you what I'm talking about. So let's start at leptin. Okay, now I showed you the mouse. And it was at this point that my current head of department, Stephen O'Ratley, actually was thinking, if we have a mammal, okay, a mouse, with a mutation, that was a naturally occurring mutation, it wasn't a genetically modified animal, that can actually result in severe obesity because of a mutation. Is the same thing true in humans, okay? Or is it some weird mousy thing? It's entirely possible it's a mousy thing. Mice are small, has whiskers and a tail, and what have you. The bottom line is him and his, at the time, clinical fellow, Siddharth Faruqi, started looking, and the bottom line is it's not a weird mousy thing, okay? Because they then found the first 2 human beings with homozygous mutations within leptin, and so they in effect lack any leptin. And leptin, as it turns out, is produced from fat, and it circulates in the blood equal to the amount of fat you have. So the more fat you have, the more leptin you have, and your brain senses this to say, okay, this is the amount of fat that I actually have, okay, for that. So what happens when you don't have leptin? You can see these kids are severely obese. Now, although they're of normal birth weight, so they come out normal baby size, they then become hyperphagic after weaning. Now, hyper for more, phagic for eat, okay, so hyperphagia, but it's I have to say, pathogenic term. So you can't say, oh, dude, I was hyperphagic last night, You ate too much. So in terms of, but hyperphagia is a pathogenic term in the sense where these kids have to have their freezer doors locked up because they'll actually go in and eat the frozen fish fingers. I'll come back and I know it's a bit until I get to the second part of this. Okay, so we'll get there in a second. So it's abnormal feeding behavior. Okay, no one would call this when you actually hear this is not a normal thing to do. Now they won't poison themselves. They're not going to eat a chair. That's a different disease. Okay, but they will actually eat food that to us might be unpalatable, although not poisonous. Okay. Now there is no defect, as we understand, in energy expenditure. You can read all of this. There's a lot of fat, normal height, but they don't undergo puberty Okay, and they have a bit of an impaired immune system. It's kind of a wonky immune system, which seems to be two odd things to be talking about within an obesity talk. But as it turns out, these last two phenotypes really begin to illustrate the role in which leptin plays, the biological role of leptin. Now, leptin is a fat hormone. Okay, so let's have a closer look at this kid. So this is what happens when you don't have leptin. This is the 3-year-old kid who's 42 kilograms. Okay, so none of this work is mine. I just want to be to be clear. So this is work done by Steve Ratley and then and Sadaf Faruqi. So I'm 75 kilograms. Okay, just to put things in perspective. So this is a 3-year-old who's 2/3 my body weight. All right, so this is not, sorry, too much cola type of, I haven't gone out to jog too much type. There's something wrong with this kid, and he lacks leptin. Okay? Now, because leptin is a hormone and circulates in the blood, and he lacks it, so Steve was actually thinking, well, hang on a second. If we take the analogy of type 1 diabetes, in which you don't have insulin and you can inject yourself to manage your glucose homeostasis, well, can you inject yourself with leptin to actually manage your fat homeostasis? This was the hypothesis. To cut a long story short, to everyone's amazement, this is the same child, okay? 4 years later, 7 years old, now weighing 32 kilograms. Okay, the only difference, okay, is that he is now so A, you wouldn't look at this child in the street anymore because he looks perfectly normal body weight, and he's just injecting himself daily with leptin. Now, at the time, the patent was held by the Rockefeller University, but they had licensed it to the American pharmaceutical giant Amgen. And oh my goodness, to be a fly on the wall, you know, in that place, when they must have thought all their Christmases had come in one go, that they solved obesity. You know, wow, we have so, but they hadn't, okay, because this work here, and you can see it down there, was published in 1999. Okay, we are 20 years on. No one has heard of the magical leptin cure for obesity. Okay, and it's not like Amgen didn't try. In fact, Amgen, when they realized this, okay, then began to sell it on. I don't even know who owns leptin anymore. But it's not like Amgen didn't try, right? They gave it to men, to women, to people of different colors, to old people, to young people. And this was the universal truth. If you had a functioning leptin system, it didn't matter whether or not you had a BMI like Kate Moss, you're a supermodel, or you looked like Santa Claus, okay? If you had a functioning leptin system, you did not respond to exogenous leptin, to additional leptin, certainly when it came to food intake. But the question is why? Because if you didn't have leptin, you can see that this child was exquisitely sensitive to leptin. Now, as it turns out, we had to then begin thinking, but all of us were bamboozled. We were thinking, well, if leptin comes from fat, and the more fat you have, the more leptin you have, surely it's a negative feedback loop, okay? Where the more leptin you have, it signals to the brain to eat less, you lose fat, okay? That kind of thing. And almost this could be a proof of principle until you think about it. Why? We never had enough food as human beings. 30, 40 years ago, we now have too much food. But we never had enough food. Why would we have evolved a system to actually inform us that we actually had too much food and too much fat? We wouldn't have. To cut a long story short, leptin does not function when there's loads of it to tell your brain that you're too fat. Leptin functions when there's too little of it. Because when there's too little leptin, you have too little fat. And when do you have too little fat? When you're starving. Okay, so leptin's role is to inform your brain when you're starving and to turn on the starvation response. What is the starvation response? The starvation response is clearly to eat. All of us know that when we are really hungry, the simple foods taste the most delicious, okay? You know, some rice, some cheese, some bread, okay? But the fuller we become, the pickier we become, okay? So we understand it's phenomenon we go through every single day. But imagine if you were starving, actually starving. Plane crash in the Andes, your partner is looking delicious, starving. Would you eat frozen fish fingers to keep yourself alive? Yes, of course you would. All of us become hyperphagic when we are actually starving, and the reason for that is because our leptin levels plummet and we begin to say, I need to eat. Okay? The other thing which your brain begins to do is it begins to save energy, okay, triage energy for the brain, so that even in your haze of hunger, you're still able to put together a strategy to look for food. Okay, it's the command and control. Now, your brain is 2 to 3% of your body weight and volume at rest right now, but it uses up 25% of the circulating glucose in your blood. Okay, so it's hideously expensive, metabolically speaking, okay, but that's because it does all the command and control. So it begins to shut off, while important, immediately unimportant metabolic functions. And one of those functions is reproduction. Okay, ladies, okay, that time of the month, loads of wasted calories. When you're at the edge of starvation, Those wasted calories are the difference between your life and death. So your brain just shuts it off. I mean, and secondly, if you're actually starving, are you going to really plop a baby out into the Serengeti? You're not going to be able to actually feed the baby at all. So your reproduction goes off. And the other thing which is very expensive is your immune system. Okay. Now you might think, well, I would die of an infection. Triaging, guys. You're going to die of hunger before you die of an infection, so your brain just kind of tunes everything down. But it's entirely reversible, because when you give leptin, you can see that the kids become skinny. It's fixed the hyperphagia, okay? Reproduction-wise, his older cousin, so it was his cousin in the previous picture, she's now in her 30s. Remember, this is 1999. Okay, she's just carried a baby she has a young family. Okay, so the first leptin-deficient human being in all of evolution to carry a baby to term because of daily injections of leptin, and immune function fixed. That is leptin's role. Leptin's role is there to actually turn on the starvation response to keep yourself alive. Now, I then mentioned, well, okay, everything else is going to be in the brain, okay? So what happens when leptin hits the brain? Well, amongst other things it does is it tickles a population of neurons called the POMC neurons, okay? Now, I can show you a picture now of children with POMC mutations. It was found by a German group in Berlin, and they would be just as obese as this child. I can show you pictures of the mice in which we actually created knockout mice. It recapitulates this, but I thought I would show you some of the more recent stuff that we did, which is on Labradors. So pet Labrador. Now, I don't know. Are there any Labrador owners in the audience? No. Okay, there's some Labrador owners. Hello, Labrador owners. So look, Labradors are the most popular pet dog in North America and in Europe. Okay, that's because they're lovely family dogs. They have a wonderful disposition, but they're also very food motivated. For those of you who have Labrador, keep your compost bin shut. Otherwise, they'll eat all the right. Okay, good. So don't do that. And they literally will pop. And so they're hugely food motivated. So this study was led by a veterinary surgeon colleague of mine, Eleanor Raffin. So she did her PhD in human genetics, but then went back to becoming a vet and was always interested in why these dogs were so food motivated. To cut a long story short, a percentage of Labradors have a deletion in POMC. Okay, not all of them, and this will become relevant in the next slide. But what you see is because not all of them do, you can actually do what we call a genotype, which is your genes, versus a phenotype, which is your physical characteristic, correlation. And you can see that, for example, if this is the wild type, of which I'm not, this is the wild type Labrador, these are heterozygous, one copy, and these are homozygous Labradors, and this is body weight. And what you can see is each deletion allele, which deletion copy, is worth about 2 kilos. That doesn't sound like a lot. So a homozygous dog is 4 kilos heavier than on average than a wild-type dog. But Labradors only get to around 30 to 35 kilos. So what happens there is that's actually quite a lot of dog. And the same thing is true for the food motivation in terms of if you actually plot it, okay? So what happens is as you have more copies of this gene, the dog becomes more food motivated, and then the dog becomes more, eats more, and then becomes more obese. And so these are the scores on the door, which I was telling you about, 2 kilos per deletion allele, and that's roughly 0.3 of a standard deviation of body weight. But so far, so not so interesting, because I already told you that humans are obese, and mice are obese. Surely dogs would too, okay? So there is a kind of ending tail to this. So about 20 to 25% of pet Labradors have this mutation. The thing is, 95% of Labradors are really highly food motivated. So the question is, what's happening to the other Labradors? And Eleanor is in the process. She has a whole team working on why this might actually be the case. But the other thing about Labradors is that they're very trainable. In fact, they're so trainable that they're primarily used as guide dogs for the blind. Okay. And they're trained, and they're like the Navy SEALs of the dog world, right? I mean, they're trained within an inch of their life because they're about to be handed a human being to look after. And when you actually look at pet Labradors, Labradors, successful guide dog Labradors, 80% of Labrador guide dogs have this mutation in POMC, making them more food motivated. So here's the concept. So here is the guide dog, okay, bringing back visually impaired Mr. Smith, okay? Now, if a chicken runs across the road, okay, what are the chances of a chicken dinner? 50-50, 80-20, who knows, okay? It doesn't matter because the successful Labrador is trained that he will have a 100% chance of a hot meal or cold meal, dry meal, okay, when he gets Mr. Smith home, okay? So suddenly we got to a situation, because that's what they're being trained, because that's what they actually have with food, and there's a numbers game for them. And so you end up with, we started with food motivation and ended up with something as trainability. So, when you have your, look, Fido loves me. He doesn't love you, he's hungry. Yeah. That's terrible. Terrible. Anyway, so this is Eleanor who leads the study, and this is Jasper, one of the participants. Okay, and it was so I am telling you the story in the Quentin Tarantino fashion. I'm telling you in terms of what happens in the pathway rather than chronologically what happens. And in 1998, when I first joined Steve Ratley's group, we then identified the mutations in the melanocortin-4 receptor, which POMC signals to. So leptin receptor, I didn't tell you about that, POMC, MC4 receptor, feeding behavior, okay? And so now you should be unsurprised to see that actually when you have a mutation in the melanocortin-4 receptor, you end up also with severe obesity, which is what we found. But what we also found is that a couple of interesting things about the melanocortin-4 receptor. First of all, it's far more frequent. So we now know that 1% of people above a BMI of 30 will have demonstrably pathogenic mutations within the MC4 receptor. Okay, and 1 in 1,000 in the general population, okay? And because there's so many, we can then actually do studies on them, right? And we can ask how functional they are. And so, unsurprisingly, some of them are gonna be big, gigantic mutations, deletions, big, catastrophic mutations are gonna be entirely dead mutations, right? Dead receptors that don't actually work anymore, whereas some are partially functional. You can get them 25% functional, maybe 70% functional, what have you. Now, so this work was done by me when I was a young postdoc in a lab with recombinant protein in the tissue culture hood. This study was done by Siddharth Faruqi, my clinical colleague, who then did this work in a clinical research facility. Never the twain did meet. I never saw any of these patients, okay? And she measured ad libitum food intake. So at a buffet, how much food you'd actually eat. And you can see here in kids, it's difficult to do this in adults, but this is done in kids. And you can see this is leptin deficiency. This is leptin deficient kids. They eat a lot. Here's treated leptin deficiency. You can see that there's a real huge drop-off. And these are the kids with MC4R mutations. Now, she tested all of the kids with MC4R mutations together. She didn't know this study, the results of this study. She only separated them out into these different graphs after I provided studies done in a test tube. And what the amazing thing was, it wasn't a binary effect. It wasn't like you had MC4 receptor or did or didn't. Okay, the quality of the MC4 receptor, is it 50% functional? Is it 70% functional? How many copies were actually on the surface could predict how much someone would eat, a child, in a buffet meal. And so it's actually a rheostat system which is there to actually tweak our feeding behavior as we go on throughout the day. And lest you think this is a weird human-specific thing, it isn't, okay? Because for example, we now know that there are missense variants, okay, the MC4 receptor in pigs, particularly pigs that are actually used for the production of back bacon, because what happens is they grow quicker, they do what they want, they don't have to be given growth hormones because of a genetic mutation, they're genetically just more likely to gain weight, okay, in a natural fashion, whatever you might want to make of that, okay? Blind Mexican cavefish. Okay, now, what are you talking about? So cavefish, okay, so these are the river fish. Not so interesting because they look like a fish, okay? But these are fish which, for whatever reason, have been swept into these underwater caves in Mexico, and in which there's no light, so they've become blind, and B, there's very, very little food in there. Every so often, maybe during a big storm, there's going to be some food that actually goes in, and there are fish in there and surviving on nearly no food, but very, very little food. But what happens is, now people know that these fish have a mutation in the MC4 receptor, okay? So when there is food available, they eat every scrap of food they can actually flipping fine, okay? So that they are able to actually pack on fish fat, okay, in order to keep themselves alive. So humans, okay, mice, I haven't showed you any data in mice, pigs, okay, and blind Mexican cavefish, okay? Now you might be thinking, ooh, these are, at least we're talking about large creatures here. No, not even that, okay? So I have a fly arm. I don't have a fly arm. You know what I mean, okay? I don't have a fly arm. Can we make fruit flies fat? I don't know if you're gonna, so these are the kind of fruit flies, these are not bluebottles. They don't fly around poo, okay? So these are the type of flies that appear when your bananas have gone brown, okay? In fact, the guys in my lab, I've brought, so you can see the thing. So I've actually brought a fruit, and you can see them flying around, and they're there. So the two glass containers, you can come up and look at it after, okay? So the two glass containers contain a high-fat diet, even for flies, I know, okay? They don't become like a little blob of fat, okay? Because after all, they have an exoskeleton. But they do have a larger fat body when we can actually make them fat. And these are just regular, they're all wild type. And on top of that, Okay, some of the flies have red eyes, and some of the flies have white eyes. And this shows some other genetic markers which are there, which you can then use to do some genetic tricks. And what we've begun to realize is you can actually take these and actually do some functional analysis on it. So for example, this GIF over here is, are the flies participating, as you do, they're not volunteers, but obviously, in the cafe study. So the cafe study is a capillary in which you fill with a mix of yeast, crushed up yeast, and sugar. Okay, so it's yeasty sugar, fluid. And then there is you just put a little bit of dye at the top, and then you can't do one fly. The flies are dead fly, it's very small. So you have to put a bunch of flies. And then you then see the meniscus drop over a period of time, right? So you know what the volume is, so you can actually, it's a cafe food intake. And so here, if you take flies, this is the normal diet, just normal sitting around doing flies, okay? These are flies that have been fasted for 24 hours and then exposed to the cafe. And you can see they suddenly eat the food at a greater rate, as you would, even in a fly, okay? And then when you put them on a high-fat diet in the glass container, they're going, oh, dude, okay, here's more sugar, here's more yeast. They eat slightly less. Even in flies, the moment people begin to think, oh, feeding behavior, no, it's habit, it's bad habits, it's morality, you're making a bad choice. I'm sorry, what morality does a fly have, okay? I mean, we're looking at really, really hardwired things. Then there's some other cool things we can do. Now, as it turns out, flies have a translucent stomach. And so we can do this study called the green stomach assay. And the green stomach assay is one where you fast the fly for a bit, so they're really hungry, right? And then you kind of put them in, but you color the food green. And so you just have to look at the flies and see how green is your tummy, right? And so you can do where here's the normal diet. Now, you clearly can't do too much of a fast, but you can do the high-fat diet. And when you make them full, they have less green tummies. Okay, you can actually do, and then you can do other things. I won't mass, clearly it's just body weight. You can do glucose levels, even in flies, ladies and gentlemen, that's right. Okay, and you can do climbing assay. Now I won't do it here, okay? But as it turns out, okay, unsurprisingly, when you're fasted, you move more, and you move more because you're looking for food, okay, pretty much. So you take the tube, I won't do it now, and you go bang, okay? And all the flies drop to the bottom because they're flies, right? And then what you didn't do is there's a midway mark, and you climb. How quickly does the fly climb up to the midway mark? And this is what happens when they're just sitting around, they're eating normally, they move. When they fast, they really climb up that tube really, really quickly, and then put them on a high-fat diet, they do the same thing, even in flies. And this is my point. My point is in flies, in blind Mexican cavefish, in pigs, in dogs, in pigs, in pigs and dogs, and whatever. Okay, in humans, that's the last person, yes. We all have hardwired tendencies to behave around food. It is not simply a matter of bad habit, ladies and gentlemen. It's not simply a matter, there is an element of choice. We'll get to that in a second, clearly, okay? But there are going to be conserved pathways where you can actually see what is happening in this situation. And right now, we're in a position where when we find new genes, we can then ask the question, well, what happens when we actually, gene edit it out of a fly and then measure them and say, well, does it change or not, okay, from a scenario and learn something more about the gene. Okay? And so this is a situation where this is now a flattened area of the brain which we're talking about, where leptin signals to POMC neurons, which signals to the MC4 receptor, which is over here. But we have a tendency, like I just did for the first, for most of the past 50 minutes, in talking about these neurons as one big group, like smoothies, okay? Here's a POMC neuron, okay? Here's an MC4 neuron. When actually, as we're beginning to do more and more work with this, it's clearly not the case, because they're more like fruit salad, okay? So clearly how pink they are depends on how many strawberries are in there, et cetera, et cetera. And so now we are doing what we call single-cell sequencing of both mouse and human donor, human dead people, just want to tell you, okay, the hypothalami, in order to find out more about the brains and more about the circuitry, in order to actually understand more about why people behave differently around food. Now, single-cell sequencing, the problem with that is you only get a whole lot of stuff in there. It's like having the names of all the countries and cities without any map. And so the other thing which we're doing is we're then taking some of these genes we identify and actually map them back into the human hypothalamus. And you can see here, for example, 2 different genes in which each of these purple dots is one copy of a RNA, okay, of the transcript, and the green genes are another gene. And you can say, okay, this particular neuron expresses, 15 copies of the purple gene and 2 copies of the green gene, and we're in the process of going through and actually mapping this out on the hypothalamus. So in the last 5 minutes, I won't keep you here for too long. Again, we want to have a discussion. Everything which I talked to you about in the leptin-melanocortin pathway to date has been what we call monogenic syndromes, Mendelian syndromes, meaning Mendel and his P's, where single genes cause a specific phenotype. Okay, these are catastrophic mutations that cause a phenotype. But look, when we're talking about body weight, and it's quite clear that the vast majority of us, okay, will not be skinny, large, fat, what have you, okay, because of single mutations, but because of very, very, very subtle variations in hundreds and hundreds of genes. In fact, now we have a general idea of the genetic architecture of common obesity. So this is from what we call genome-wide association studies. Now, you're not supposed to look at all the genes here. A couple of messages I want you guys to take away from this Jackson-Pollock diagram, okay? So what happens is, you'll see that there are two broad groups. There's a pink group up here, and then there is a big blue blob that's actually down here, okay? So the pink group up there, as it turns out, are genes which actually play a role in your waist to hip ratio. Okay, so this is the circumference of your waist over the circumference of your hip, and if you see that as a ratio, you see its body shape. Do I have a big tummy and a small bum? Vice versa, do I look like a sausage, right? So it's, and as it turns out, it's where you put your fat. So these are genes that have a role in where you put your fat, okay? These genes here in the big blue blob have to do purely with your BMI, independent of where you put any of the fat. So first piece of information I want you to take away is that the genes about where you put your fat, at least genetically, has nothing to do with how much fat you have. Okay, so this is the first piece. These, as it turns out, okay, so that's the first message. The second message is these genes play a role in where you put your fat, as it turns out, has a role in fat biology. So it sits in your adipocytes, you know, and so where you put your fat has to do with your fat. That sounds like it makes sense, right? These genes, however, with your BMI, the vast majority of them are enriched within the brain and function within the brain, and where we know their function influence food intake and feeding behavior. This is another way of looking at the data. This is called a Manhattan plot. And it's called a Manhattan plot because it looks like the skyline of Manhattan. Now, I have spent 12 years of my life, count it, ladies and gentlemen, 12 years of my life working on this gene called FTO. It's a very painful experience. I won't share it with you, okay? Except to say that half of everyone in this room will carry one copy of the FTO variation, which makes you, on average, 1.5 kilos heavier than the person next to you who doesn't have it, okay? 1/6 of you will have 2 copies of it, which makes you 3 kilos on average heavier and 50% more likely to become obese than the person next to you, okay? So these are not mutations, okay? All of you will have some mix of these genes that are here, of this individual mix. And what genes are here? I've highlighted 2 of them because we've talked about them. Here's the MC4 receptor, blind Mexican cavefish, the pigs, blah, 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 okay? And here's the Labrador, the POMC, okay? So remember, these are not mutations, these are very subtle variations. When you have very severe mutations, deletions, et cetera, you have a severe phenotype, as I've been showing you. But when you have very, very subtle changes in these genes, what ends up happening is it influences your where you sit in the normal distribution of body weight. Okay, so you can actually create a risk score. So we now know of hundreds of genes. But imagine we just take 100 genes. Okay, so you can have there are 3 possible scores for each gene. You can have the risk copy, 1 risk copy, 2 risk copy, or no risk copy. So 1, 2, or 0. So you have if you take 100 genes, that's a notional minimum risk score of 0 and a notional maximum risk score of 200. Okay. But that's not how it works because this is not how biology happens. And so all of us have an average number of these different alleles because that's the way biology works. Our different mix is independent mix, but we all have roughly the average. But some people will have more of these, and some people will have less of these. And if you actually plot the number of the risk alleles you have, the risk variations you have, against BMI, what you see is a linear relationship. So the more of these variations that make you slightly heavier that you have, the more likely you are to be heavier, okay? So people always ask me the question, well, does it mean that we can actually find people that are sitting out there and actually make a prediction that, like a baby, and say, this is a person likely to be obese, let's intervene now, okay? And then, people want to do this, right? We can actually tell and predict the genes. I'll cut a long story short. The answer is we can't yet, okay? For any number of different reasons. It doesn't stop people from doing it. So this is, look, I live in a little tiny village just outside Cambridge. We have 2 windmills, one of which doesn't work. We have 2 churches, one of which is deconsecrated, and a pub. I can't buy bread in my village. But someone comes along and slides this under my door. I could buy a genetic test in my village, though. Okay, so this is from 23andMe. Other tests are available. Now, look, they are perfectly capable and fine in terms of saying, is there an A there, a T there, a G, whatever. It's going to be fine, right? But what they're terrible at, all of the companies that are out there, is actually interpreting the data in any useful sense whatsoever. Fundamentally misunderstanding the difference between population-level risk and individual prediction. Okay, and there's just a huge gulf of difference between looking at population-level risk and saying that I predict that you're actually going to actually have something. Hopefully we'll get there at some point, some level of prediction, but ladies and gentlemen, just don't waste your money at this point. So do I give people an excuse? I have 2 more thoughts and then I'll shut up. So do I give, look, I think you've got to consider your genes, not the monogenic Mendelian type. Okay, we're talking about a regular, normal type of any human complex trait, like a hand of poker. Okay, you can have good hands, you can have bad hands, you can blame your folks. That's the only people you can actually blame for your genes, right? But you can win with a bad hand of poker, even though it's more difficult. I'll give you another example. I will never, ever be able to run as fast as Usain Bolt, okay? And it's because of my genes, and I'm sticking to that, right? But that doesn't mean that if I train, I won't run faster than I actually do now. What your genes do is not give you a position. They're not deterministic. But your genes do bracket a level of possibilities. Why am I bald? Because of my genes. Why do I look Chinese? Because of my genes, right? That's just, they bracket possibilities. But within those possibilities, you can move, okay? They're not deterministic. They're just there to give you a range of possibilities. So, actually, now I have 2 final points. I don't know if you guys ever considered the dessert tummy before in your life, the dessert tummy, all right? So the dessert tummy, okay, I'm sure we all know, is when you go after maybe you haven't had tea, you're going to go for your dinner. I guarantee by the time you reach your, as long as you go to like a proper restaurant, not some Michelin star small food, okay, like regular food. So I guarantee you, by the time you reach the middle to the end of your main meal, you have achieved your metabolic requirements of the day, meaning that you've recouped the calories that you've spent for the day. But yet the chocolate cake comes and you eat the chocolate cake. Okay, so this is the dessert tummy. The interesting question to ask is, why is it specific for chocolate cake? Say you've gone out for steak and chips or spaghetti, whatever it is, okay, and you finish your meal and you're clearly full. If the waiter comes along and asks the question, more steak, more chips, you can, oh no, dude, I'm going to puke. No, you won't, right? But yet you'll eat the chocolate cake. So what is specific about the chocolate cake that actually makes you eat it? Okay, so once again, evolution. Let's hark back 50,000 years ago in the Serengeti. This is an antelope, okay? So now imagine it's taken me 2,000 calories to take down the antelope. So what happens when I get back to the village? I have to eat at least 2,000 calories, right? Otherwise, it's just a bad idea. But if I only ate 2,000 calories, there's no guarantee I'll get the antelope the next time round. So what happens is a part of the brain which really, which makes eating feel nice, the chocolate cake feeling, okay, takes over so that you eat more than you need, okay? So this is, the question is, how do you get past the mechanical problem of being filled with 2,000 calories worth of venison? And this is the question. Well, your brain begins to change the type of calorie that it begins to want and begin to like. It begins to increase the caloric density of the food that you begin to eat, which means that for every given gram of food that you eat, you can get more calories. So you can actually stuff them all into all the little nooks and crannies that are actually in there. So what is high in caloric density? 50,000 years ago, there would have been foods high in free sugars, so ripe fruit, maybe some honey, okay? And obviously fat. What is high in sugar and high in fat? Desserts. So your dessert tummy is a holdover from your time in the Serengeti to make sure you could continue eating as much as possible, okay, even when you were stuffed to the gunnels, so that you could prepare yourself for a famine that's not going to come. Yeah. And happy dinner, guys. Unless you think that this is some weird human thing, once again, okay, this is the thing. It's not. Consider the grizzly bear. Okay, the grizzly bear is not going to be having a muscat with his Tao citron. Okay, I realize this. But consider the grizzly bear hitting the salmon run in the Pacific Northwest getting ready for hibernation. Okay, so they go there clearly to gain as much weight as possible. The grizzly bear, when it starts, will finish the entire salmon bar down to the bones, tuk, 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 right over here. But as they get fatter and fuller and fatter and fuller, the grizzly bear only eats the skin and the fat underneath the skin. It begins to increase the caloric density of the protein is dense in a certain way, but not in terms of just pure calories. So it's not dessert. I appreciate this. But the phenomenon is exactly the same as our dessert tummy. So are we all sinners? Okay? Because I hate it when we talk about sin, because when we talk about sin, we talk about choice. Okay? And this is where we're and this, by the way, is me choosing to eat a pizza, right? So we actually are talking about choice. And look, as it turns out, people who have a genetic tendency to actually put on weight, people who are going to be heavier, because of biology, find it more difficult to say no. You have to remember, because of their genes within their brain, find it more difficult to say no for a myriad of different reasons. Now look, okay, clearly, you have to remember that a couple of things. Clearly, it says, when I talk about obesity as a choice, yes, it's a choice. I'm putting this pizza or cookie in my mouth. I'm eating the pizza. It's a choice, okay? And this is actually very, very true. But we do not gain weight overnight, and sadly, neither do we lose weight overnight, all right? But our body weight, as it stands today, is a function of thousands of different food decisions you have made over the past months and years. If your genes make it slightly less likely for you to say no, 5%, 2%, 10%, 3%, whatever the number is, then over years, no matter what you try and do, you are always going to be slightly at a higher risk of becoming heavier than someone else. Ladies and gentlemen, obese people are not morally bereft. They're not bad. They are fighting their biology. And even amongst this obesity epidemic, whatever word you actually want to use for it, if we as a society cannot accept this, okay, and we push the blame back on the people suffering from the problem, I draw a huge distinction between pointing out the problem, which there is one, and blaming the people suffering from the problem. Until we accept that we can't blame the people suffering from the problem, we are never, ever going to fix this problem. So look, this is me choosing to eat the broccoli. Okay, so I just want to thank a few people before I shut up. My current boss, head of department, Sir Stephen O'Reilly, Tony Call, who I've been, and some of my team who've been with me forever are here. So Brian Lamb and Marcella Ma are here, there in the front row. Lorraine sadly couldn't be here, but she provided the flies. Eleanor, I told you about with the dogs. A number of the other guys are here. I think Georgina is here, Natalie is here. I hope they're here somewhere. Okay, in here. But last but not least, this is a picture from my wedding. Okay, a while ago. That's my dad. My dad came all the way here just for this, by the way. I don't know where. So he's actually sat there. Oh, that's. In fact, my dad just to give you an idea. So I have this book. My dad read the book twice, and not only did he read the book twice, he can now quote it. He's like evangelical now about it. He can quote it chapter and verse. He even showed me yesterday that he says my index is lacking. Okay, he's created additional index points. He says, FTO, where's FTO? It's not in here, and so he's created between the, fat Labradors and something else, there's FTO now, page 48. So anyway, and clearly, A, I'm thankful for him being here. B, thankful for him raising me in some sense. But most of all, I want to thank, my son is also here, but I want to thank, this is my wife who's also here, over here, both enjoying a drink, and without whom none of this, the love of my life, without whom none of this have been possible. Thank you very much, ladies and gentlemen.